And welcome to today's episode of the BioWaves podcast, where we have the pleasure of hosting Dr. Feldman, a distinguished professor of neurobiology and the Coates Family Endowed Chair in Neuroscience at the University of California, Berkeley. In addition to his research, Dr. Feldman serves as the Neurobiology Division Head for the Department of Molecular and Cell Biology and directs the T32 Neuroscience Training Program at UC Berkeley. He is also dedicated to teaching undergraduate and graduate courses in circuit systems and neuroscience. Take it away, Dr. Feldman.
All right, terrific. Well, thank you for asking me to meet and chat with you guys. Actually, let me start off by making a few updates on the little intro that you gave. So we recently founded a Department of Neuroscience on campus, and so I'm no longer in the Department of Molecular and Cell Biology, but now I'm in the Department of Neuroscience. And I'm a professor there, and I'm also a chair in that department. So yeah, we're very excited to bring broad neuroscience to undergraduates at Berkeley through the neuroscience department. We have a really broad department that spans, all the different aspects of brain function, how to understand the brain, how it works, how it goes wrong in disease. And this spans across really multiple levels, from the molecules and the cells of the nervous system to kind of the circuits and computations that it performs, to how the brain mediates behavior, to how the brain underlies our internal mental lives and our cognitive world, and the computational principles for how the brain works, kind of all in one department. And within that broad range, what my own lab does is we study the function of the cerebral cortex of the brain from a circuit and systems perspective. That means, how is it that neurons are wired up together into small circuits that perform kind of elementary computations? And then on the systems level, that means, how do large groups of neurons work together to represent information, process information, mediate important functions like learning and attention, things like that? And then how does that underlie behavior, and particularly our sensory perception and sensory behavior? So that's what my lab studies. We study that in mice using touch as a model system.
Thank you. Would you like to prepare do you have like a slideshow that you would like to show regarding the research, or do you want us to just take it away with questions now?
I think let's just do questions.
Okay, cool. Do you want to go ahead and start with the first one?
Yeah, I'd like to start off by asking what inspired you to pursue a career in neuroscience, and how did your academic and professional journey lead you towards specializing in your field?
Well, neuroscience has been a very exciting field for many, many years. I was an undergraduate a long time ago, but even back then, neuroscience was, an area where a lot of discoveries were being made. It was very clear that there was a tremendous amount that was unknown, so it seemed like a very exciting place to do biological research. And I had always been interested in behavior and computation, and the brain combines both of those two things. How do you have this organ inside your head that mediates your feelings of the world, your behavioral choices, your decisions, your memories? And even though that's a biological organ, it is performing computations on information about the world to make those choices, to make those processing steps. And so even way back when, it was very clear that there were really interesting biological and computational puzzles to solve about the brain. And when I was an undergraduate, I actually got involved in undergraduate research, like perhaps you guys or many of the people listening. And I got really excited about doing research on brain function, and that's really what got me turned on.
And would you be able to share any sort of experiences? I know you said you worked in an undergraduate lab with maybe mentors or any other pivotal moments that had shaped your approach to research in present day.
Well, that's a good question. Over the years, I've had many mentors myself, and I've also mentored many people, and I've known many, many faculty who mentor undergraduates and graduate students and postdoctoral researchers. And from that, I would say that there's really many different mentoring styles and many different ways of doing science that really engage the mind and are exciting. There's no one mold for doing it. And that's one of the great things about science, that science is this endeavor that we as a field have shaped into being something that is, over the long term, pretty effective at figuring out how things work, right? It makes mistakes along the way, it recognizes them, it usually corrects them. So it's fits and starts, but it really does work. But the amazing thing is that that process works despite the fact that the people who do science are all individuals, with their personalities and their goals and their different interests in life and different ways to approach a problem. And when you interact with people as mentors, you see all of those different styles and goals that people have. Um, so I can't, so it's very broad. My undergraduate research mentor back when I was an undergrad was a guy named Mark Baer that was at Brown University at the time. He's moved on since. But he had a great knack for thinking about biological brain function and what simple mathematical or computational rules the brain might be pursuing to underpin, essential functions of what the brain does. And so he had a knack for kind of simplifying complicated problems to think how they might map onto simple mathematical equations for what the brain might be doing in some particular circumstance. And I found that very engaging that one could make headway on studying a complex problem by thinking about it from simple frameworks. So that was very inspirational.
Yeah, I mean, wow, it's pretty like inspiring. Like, uh, just to know like how much he shaped you, or not shaped you, but like that sort of philosophy imparted onto you. And I mean, I can see that there's some like a lot of wisdom in like what you were saying about like all the different like philosophies and approaches different people have different mentors like take. But like, you were like saying like a lot of our audience and like us ourselves, like we're students, we're also looking to pursue this kind of research. So like what sort of like guidance or like advice would you offer to like students or like maybe even like early career researchers who are trying to like make a meaningful contribution like in neuroscience?
Um, well, when you're starting out, you're trying to do so many things, right? You're learning how to work with your hands typically on some kind of experiment. So you're learning methods that you've never done before. And you need to learn how to be extremely careful and observant and reproducible and all the things that you need to be with your method. So you're thinking about that. But then you also have been given some project to work on, and there's typically an interesting intellectual history behind that project. Why is that the question that your graduate student or professor has asked you to work on? If you succeed in making those measurements, how will your results be interpreted in a way that really adds to the field, changes how people think about the field? Um, and then, of course, you need to learn how to analyze your data and interpret things correctly and all that. So you're doing many things at once, and all that's exciting. And I would say people get excited at different levels of that. Some people, love working with their hands. Some people love troubleshooting. My method doesn't work. How can I figure out what's going wrong? Some people are not so excited about the actual hands-on part, but they just love thinking about the ideas of science. And you need some combination of all of those, but you don't have to solve them all at the same time. So I'd say when you're starting off, let yourself get excited about whatever level is the most appealing to you. But the thing that you want to do at that level is you want to immerse yourself in it, right? You have a unique opportunity as undergraduates at Berkeley or people, undergraduates at other great institutions, to really do important, meaningful, cutting edge research where no one knows the answer, and you're going to help discover the answer. And my advice would be let yourself get into that. It's exciting. It's fun. It's also hard work and frustrating, right? But
Yeah.
let yourself get into it.
And more on that topic as well. Obviously, science is rapidly changing, and I guess my next question on that would be, how do we reconcile or say navigate those rapidly and the rapid advancements in, for example, neuroscience? Like, how should we continue to stay up to date with this landscape?
Yeah, that's a complicated question. I think um, for the people who are already in the field and are really trying to do novel experiments, independent research. So people beyond the undergraduate level, but maybe like PhD students or postdocs or faculty. it has become a major challenge in the past 10 years just to keep up on all the papers that are being published in the field related to your work. The volume of publications has gone up tremendously. So I think one of the challenges is becoming adept at reading broadly and digesting information in ways that you can build up models. And then your goal as a scientist is to kind of understand the competing models that are out there, and then either to do experiments to prove or disprove them, or to come up with your own better model and try to prove that. So you do inescapably have to keep up with, all that information in the field. It's different when you're just starting. It shouldn't be your job, I don't think, as starting undergrads to be an expert on everything in the field. It's too much. You have wonderful filters, right? You have the grad student you're working for, the postdoc you're working for, and they can really help you develop an intellectual framework that for thinking about your problem that is simpler than all the papers in the field, but references the key papers in the field. And that's what you should really try to keep up on, not every twist and turn. The other aspect about it is technological. Another thing that's changing in neuroscience all the time are the technologies that we can use to investigate brain function. And actually, Berkeley is a great place for this. There are so many faculty here who develop novel methods through engineering, molecular biology, physics, all sorts of different approaches, in order to be able to ask new questions about brain function. And so in those labs, it's often that, half or more of the lab members, their job is to build technology, develop technology, test it, try to prove what it can do. And then the other half of the lab maybe is applying that to some biological problem that they're interested in. So another challenge in being in science is keeping, in a way, kind of mentally young enough to be able to appreciate and adopt and learn those new technologies as they come online. It's another fun thing about doing science. And I would say another challenge of all this is, what about people who are trying to follow, or how should people follow who aren't in science at all? And this is a big challenge in modern science, right? If you think about the stream of information that comes on, TV or in social media or in your newspaper about recent discoveries in brain health, or, various neurological diseases, or actually, you pick your field of medicine that you want, pretty much anything. And so it used to be that all these individual advances and debates and fights and resolutions and all that were done underneath the hood of science, and only later did the major results kind of percolate out to the public, which made the pace, I think, kind of absorbable for most people. But now that's not really true. Now there's an awful lot out there that, one glass of wine is good for you. No, one glass of wine is bad for you. How much exercise should you get each day? You can name all the examples. And in a way, it's kind of nice that the inner workings of each round of scientific discovery are now more accessible to the public. But it's also very challenging, because unless you're an expert in the field, it's very, very hard to know which of those to believe, which not to believe, or maybe if two studies are believable but contradictory, what might be the difference? How do we figure that out? And unfortunately, my personal opinion is that this has been something that has contributed to maybe some reduction in the appreciation of science by the general public, being tossed one way and another with all these seemingly conflicting things that are part of the process of science, but are a little bit too much than most people want to be following all the time.
Yeah. So more on that as well. Sorry to take this over here, but also there was another question I had planned to ask. I was planning to ask it at the end of the podcast, but I think it's a perfect segue. So do you think that as a scientist, it's a responsibility to make the information that we produce more digestible to the general public in order to spark that, I guess, inspiration for people to continue researching the field in the future and to understand what kind of work we're doing? And then kind of a second part to that question, you said the rate of publications that are being produced in the field of neuroscience is, rapidly evolving and obviously becoming larger. So I'm sure you've heard of the singularity theory. For the audience, this theory assumes that there's a hypothetical time where machine understanding and intelligence will surpass humans, and therefore the evolution of technology will be expedited and grow much faster. How do you think this would influence the pace of discovery in neuroscience, let's say over 10 years to 100 years to 1,000 years?
Boy, the 1,000-year question, that's a tough one.
Yeah.
Yeah, I mean, obviously, this is the big unknown, right? There have been huge advances of AI that have been affecting, every segment of not only science, but lots of society. Of course, there's a lot of debate about the value of that, the challenges of that, the advantages of that it can bring, right? And I think, we have to go along for the ride to figure out, the pluses and minuses that are going to come along with that. My feeling is that right now it's a little too early to answer the question because you're suggesting a certain type of AI that is, let's say, trained on enough neuroscience that when asked a question about how something works, it could give an answer that maybe is as good as what a PhD trained person in that scientific area would be able to give themselves, right? So that might be the tipping point, that if that happens, maybe we don't need as many scientists thinking about the problem. I would say that we're not there yet. I can give you an example of that. Okay, this past fall, there was a paper published that I'm not going to get the exact headline correctly, but the headline was something like AI can now predict future discoveries in neuroscience. Okay, but the experiment was this: that someone trained an LLM type artificial intelligence machine, like a ChatGPT type of AI, on a large amount of neuroscience papers. What they actually did was they went onto the major publications database. They looked up every paper in some vast fields of neuroscience, except for the last year. They did not do the last year. And then they fed all of those abstracts, or maybe it was all of those papers, I don't remember, into the AI as its training data set. Okay. And then once the AI was trained up, they then wanted to test it. And the way they tested it was they took abstracts from papers from that most recent year. These were the papers that the AI had not been trained on. Okay. And they took those abstracts and they took them veridically, like exactly the abstract that was printed for the paper. But then they also took a copy of that abstract and they had experts in the field who they hired for this change the meaning of the scientific result. So, for example, if the scientific result was we gave drug A and blood pressure went up, they would change the abstract to read we gave drug A and blood pressure went down. So everything was semantically, correct, and the sentence structure was correct. It's just that the results were wrong. Okay. So now they have two test sets. They have the actual real results and they have these opposite results. And they asked the AI to judge which of those two abstracts was real and which was not. And the AI did that with some level of performance. And then they gave exactly those same abstracts to a set of faculty and postdocs with PhDs in neuroscience. This was all in neuroscience. And they asked them to make the same judgment, and they did not do as well as the AI. Now, because the AI had not been trained on those papers, from the AI's point of view, it was like predicting the future, right? And so that's why the headline is kind of like the AI can predict future results in neuroscience. But here's how I would answer the question that you asked. What's this experiment really doing? It's not really telling us that AI can predict the outcome of experiments. In my view, what it's telling us is that a lot of papers in neuroscience, probably like in any field of science, actually are fairly incremental. They're making findings that are well predictable from things we already know, right? Maybe that's not the science that is really moving the field along and making major new discoveries. Maybe the major new discoveries are the 10, 20, 30% of papers that are not doing that. They find really unexpected things and therefore they make novel discoveries. And because they're really unexpected, I would predict that the AI would not be able to predict those results. So this is my very long-winded answer for saying it may be the case that an AI that we have soon might be able to make simple predictions, and understanding in neuroscience, but are we ever going to have an AI that's actually creative enough to discover, quote unquote, something that truly has not been discovered before? I don't think so. I think that's what science is really designed to do. We're anchored in real experiments because the real experiments can make discoveries we don't expect.
Yeah, no, that's like a lot of like conversation about like AI. It's sort of like, especially with LLMs, like they at one hand they seem like they really know stuff, like they're very intelligent, but then at the other hand, like they are really in a sort of sense just like token predicting machines. Like they're just trying to like that's all they can do. Like there's not really any reasoning behind them, like in a technical sort of sense. Um, but still, like it's very like fascinating. Like even now, like the example you were giving, like it being able to like predict the direction of research. Like, do you think like if there was something like that that could sort of like find broader implications of like I don't know, like a published research, like could that have like huge like implications in neuroscience? Even something like very small like that.
I mean, it certainly could. Um, in a way, that kind of approach, but not with fancy technology, has been around for many years, right? You only had to go back, let's say, 30 years, which to you guys is a lot, but to me is not so much. And the way you learned about what was known in the field was you did literature searches in books, right? So you would look at, let's say, review articles that were published in the last 5 years, and then you would go read the papers that those review articles cited, and you would try to put together some picture in your mind about how some topic worked, right? And maybe you would learn, multiple new players in that you hadn't thought of, and then you would go do lit searches on those and put it together. And so, um, with the advent of searchable online databases, right, that we've had now for 20 years, you can very simply go and look up those terms on PubMed or even Google, right? And you can do dramatically faster what we used to do very painstakingly by hand. And so in some way, I think AI just accelerates that more. Like it's doing the looking and the cross-referencing and the putting together. Um, whether that is going to be, so that may certainly be faster than an individual scientist doing that. But whether that will lead to really novel insights that couldn't have been made by scientists, I don't know. So that's why I said at the beginning, it's very hard to predict. I'm sure that these will be useful in many ways, but it's hard to predict so many years out because I don't think we know their capability yet.
Yeah. So actually, kind of we're talking all about like AI and, it's very useful, but I want to even go straight further. Have you heard of like the new quantum technology?
Only what I read in the newspapers.
Yeah. So like it's very, very significantly faster and um it could give results at a overwhelmingly high rate. What do you think of that technology having an implication in the neuro field?
Well, I mean, if quantum computers or faster computers of any sort become widely available for people to use, then of course they'll make every computation faster. We can do much more complex things. So I'm sure it'll have a major impact. I'm not so sure what the specific computational abilities of these particular machines will do per se, but I'm sure that overall we science society has always taken advantage of um, computers and before that simpler computational machines, getting faster and more complex. We've always found ways to, use that to revolutionize people's lives. And I'm sure this will be, no different. It's interesting to talk about the evolution of computation, of engineered computational systems, because really, one of the fascinating things about studying the brain, for those of us doing neuroscience, is that the brain for a long time has been what we thought of as somehow the most sophisticated computer. Maybe not the most accurate computer,
Yeah.
right? But certainly an incredibly fast computer, an incredibly flexible computer, and one that still uses tremendously less power than I think any engineered system that's been built, right? And to discover how that works, I think is well, certainly is one of the goals of neuroscience, and I think will continue to bring advances in engineering and computer science as well. The way that we thought about this for years is that your brain, is composed of these, wet, sticky cells. It's a computer, but it's a computer that's built of fat and salt and protein. How can that possibly work? And how can that do a better job than the computer, on your desktop in front of you? But remarkably, it does in many ways.
Just to like sort of build off of that, like, are there any specific like topics or like questions that are locked behind computational power that like, oh, if we only had like more powerful computers, we'd be able to make this huge breakthrough.
In researching the brain, you mean?
Yeah, in like neuroscience.
Well, it has been a dream to try to build a cellularly accurate or maybe even a biophysically accurate model of the brain, and to use that model to see what kind of computations it can perform. I would say that we have a first pass understanding of how small circuits in the brain compute for many small circuits. We have a general theory for how large recurrent circuits like the cerebral cortex compute, but it's a very general theory. It's not really specific. These days, that theory is often stated as the brain functions like a recurrent neural network, an RNN, which is a classic type of computational model that stores memory and does many things, and is some of the hidden architecture behind transformers and other architectures that are used for LLMs, for example. But in detail, we don't really know, how most of it works. There's been a big push in neuroscience to try to discover the full list of all the cell types in the brain and all of their biophysical properties, and even more audacious, all of their synaptic connectivity patterns. One way that people have tried to do this is in model systems like a mouse, for example. Take a little piece of the brain and section it at electron microscopic resolution, image it at electron microscopic resolution, so you can see membranes and cells and every little protrusion, and that way you can find cells and you can find synapses. And over the past 10 years, many groups have developed methods, often with machine learning, to turn those 3D electron microscopy image sets into wiring diagrams of that little chunk of the brain. And so if you have the wiring diagram and you know the cell types and you know the properties of the cell types, in principle, you should be able to build that into a model, a virtual model in a computer somewhere. And then you should be able to hit run on the model and see what it does, right? That last step has proved very, very difficult. There's a famous example in Europe called the Blue Brain Project, in which they took a tiny, tiny little chunk of whisker somatosensory cortex of mice. This happens to be the part of the brain my lab studies. And tried to build that in silico in an accurate way. And they discovered many interesting little computational principles for how it's built, but they haven't really been able to say what the overall model does. And there are groups at the Allen Institute, there's groups in several places that are trying to build bigger and bigger data sets like this in different parts of the brain and the retina and other places to really figure out how it works. And so small steps, but it hasn't worked yet.
Yeah. I just had as you're talking about AI and computers and neuroscience, things like that, I just had a question. There is a technology that's being discovered where they're trying to get neural cells and they're trying to create brain supercomputers based off of neural cells and then have them run. And they feel like that's much more efficient compared to the current supercomputers we have today. They're researchers are trying to create technologies on that line. So what do you think, with regards, either on the research side or even on the ethical side for a technology like this in considering?
When you say using real brain cells, do you mean using actual real wet brain cells as part of the
Yeah.
machine, or you mean
I
designing
don't like
based on brain cells?
Yeah, they use organoids actually.
Um, yeah, I've heard a little bit about that. I don't know the details of it. Yeah, there are important questions about organoids, but I think we're probably still pretty far away from, the really serious ethical questions. So obviously what you're getting at in your question is if organoids are sitting in dishes and they're plugged into computers somehow and they're in some giant loop with, electronic systems to be part of a computer, then if those organoids exhibited features of function that we associate with normal intact brains, then we would be worried that those organoids are sitting in the dish thinking. And then that would be a huge ethical problem. In practice, where the field is with organoids is very, very far from that. So, as you know, organoids are typically created by taking skin cells and then de-differentiating them back to stem cells, and then re-differentiating them into various neuron types, which is done in an approximate kind of way, and then combining those neuron types together in some ratios that people think might give interesting properties, and then growing them in a dish. And when they grow in a dish, they don't grow into brains. They grow into little clumps of cells that are mature enough that they have some spontaneous neural activity patterns, but they don't have the structure and type of activity that you see in even very, very basic recordings from an intact brain. There definitely are people in the field who are debating how to set the right limits and recognize where the dangerous steps would be and regulate them. And I think that's extremely important that all that happens. As far as I know personally, we are very far away from that now. Now, does this mean that organoids will be useful as part of a computational system? I don't know. I don't know if they're doing sophisticated enough computation to be able to do that, or if we can interface with them at enough detail to be able to make use of that. But I'm not really up to date on the literature there. I think that's very new. I think this is something that people have been thinking about and trying in the past year.
Yeah, it was relatively around like 24, 23-ish. I think there were like news came out about it, but I'm not too sure if like was it published into a paper, what they did, how did they manage to do it? I'm not exactly sure on that.
Yeah, I don't know.
Yeah, I'm sure of you've looked at that one video where it's like the neural cells are like playing ping pong. Have you looked at that?
I heard somebody talk about that, but I haven't seen the video myself.
Yeah. So it's like I think they trained some neural cells to like train them to play ping pong properly, and eventually they like started hitting the ball and everything. It's like this is kind of like a segue to the next question. It's about kind of consciousness, and like many neuroscientists have like different takes on consciousness. Do you think we will be ever fully explain what consciousness is? Like um, pretty much that this biological mind connection kind of thing.
Well, that is the question that has been driving, much of neuroscience research, for 150 years.
Yeah.
And before that, philosophers have debated the question of how do you even define consciousness? Is it possible to study from a physical sense for even longer than that? So this remains, the greatest unsolved mystery in brain science, but it's also an incredible challenge. And I think one thing that we've learned in science is that the whole process of science is that you iteratively, piece by piece by piece, figure out your way to solve what seemed at the outset to be impossible problems, right? And there's both a scientific method for doing that, but there's also kind of an art for doing that, how you pick the questions to be able to do that effectively. So in some sense, we could, re-ask your question, not will we ever figure it out, but given where we are now, is it worthwhile to take the steps that we can to try to move down that path or not? And there, I think the answer is very clearly to me, yes. There are some great experiments out there which implicate certain types of physiological processes in the brain, and in some, maybe more limited ways, certain brain areas in having neural activity that's very correlated with conscious perception. Let's say consciousness means so many things. That's one of the hard things about this discussion. But let's take like your perception of the sensory world, your raw feeling of what you're seeing, the redness of the wall behind me or whatever. We often call that sensory qualia. So that immediate, I'm seeing a movie with these features in it perception, that's called qualia. So let's take that aspect of consciousness. There are some really quite striking studies that can tie that to particular physiological processes and particular activity in particular brain areas. Not in a causal way, that we can't necessarily do so well yet, but in a highly correlative way that at least guides us towards being able to come up with concrete neuroscientific theories of consciousness, which weren't even really possible a generation ago. So does that mean eventually we'll be able to solve it? I don't know, but it means that we can take steps towards narrowing down the problem and focusing the problem so that it may become doable.
So just to expand on that a little bit further, I did want to leave into interpretation kind of the, I guess, philosophical aspect of consciousness. Do you think that, given this description of consciousness, is it inherently biological? Is consciousness just our ability to think and reason, or is it our ability to act on our reasoning? Like, is it required for thought to be able to be put into physical change through, for example, our body, our hands? Is that what makes consciousness, or is it more of just the ability to reason?
Yeah, it's a great question. I think a reasoned answer to that needs to come from a perspective of philosophy, right? Which I am not. Okay, but I can give you kind of my neuroscientist take on it. I think we have historically thought that there's something about the brain which is uniquely producing consciousness. A person without a brain, even if they're, or without a living brain, even if they're alive, does not exhibit what we consider signs of consciousness at all. Things in the world that might behave in kind of hard to explain ways, but if they don't have a brain, they don't seem to, really have what we think of as consciousness. So for me, there's some very strong tie between those two. There's lots of interesting discussions about what aspects of brain function in a algorithmic computational sense might be the things that are most closely tied to the behaviors that we associate with, as signs of conscious behavior. And people have interesting theories for this. Many of those features of brain function are things that you can replicate in a computer network. So does that mean that a computer network with those properties would be considered conscious? This is where you need the philosopher. My gut feeling tells me no. My gut feeling is that if you created something like that, what you would have created is an incredibly good mimic of something conscious. But I can't defend that from first principles, but that's just my strong feeling.
Thank you.
Yeah, like you definitely like pretty interesting actually to like see talk like for us like to be talking about like consciousness actually like originally for me I felt like consciousness is more of like a like more of could be very much explained through neuroscience but understanding like the philosophical aspects also does play a pretty important role because like the way people think and believe like that also can like shape what their consciousness looks like as they see through like people like Sigmund Freud who talked about this who talks about like different things, the aspects like the id, superego, things like that. But I also want to like transition a little bit also onto your research, like what you I had we had all had like a few questions regarding your research. So in your work, like I read, like you did challenge like some assumptions regarding inhibitory signaling for neurodevelopmental disorders like autism. So could you elaborate on how these findings might be able to influence future therapeutic strategies for neurodevelopmental disorders?
Sure. So let me start with a little bit of discussion of how one goes about studying or even thinking about autism from the point of view of biological, research. So, autism is an incredibly broad and diverse condition. Actually, really, almost certainly, it's a broad and diverse set of conditions. And how to understand the different forms of autism is complicated. There's this idea of the autism spectrum, which gives kind of the misimpression that it's a one-dimensional spectrum that goes kind of from low functioning to high functioning, but that's not true. It's really a multidimensional thing. And one of the things that the field in general, not my lab, but other labs have learned over the past 10 years, probably the biggest set of discoveries in autism, has been that it's extremely highly heritable and therefore genetic. Okay, so there's very good estimates from a set of very large studies, well, one data set with many studies from a very large study of twins in Sweden, actually. And just by having a very large set of people tracked over long periods of time, they were able to establish that the heritability of autism seems to be 85%. Okay. Now, that doesn't mean that there's not other environmental causes, right? But it means that an awful lot of it is heritable. And then when many groups started to do genetic studies of large groups of families with different types of autism. A lot of this was done here in the US. They discovered many individual genes that were strong predictors of developing autism. And right now but there's many genes. This is a big challenge. No one gene accounts for more than 0.5%. The biggest one is 1%. Many of them are like 0.1% or less. And there's a list of about 100 of those genes. So there's 100 of what we consider high risk autism genes. Collectively, those genes, they are not responsible for all forms of autism. They are associated with what we call severe syndromic forms of autism. These are forms where one gene mutation is enough to give you the disease, and you don't have just autism, but you also have a variety of different neurodevelopmental conditions, of which autism is one part. Okay, and these are conditions like Fragile X syndrome. There's a syndrome called tuberous sclerosis. There's a syndrome called Rett syndrome. There's a whole variety of these syndromes. So when biologically we want to study what's happening so we know a lot about the genetics, but none of that tells us like what's going on differently or wrong. I'm putting quotes around that, wrong in quotes, in the brain and autism. The genetics don't tell us that. We have to do research to figure that out. And the hope for doing that research is that we can find some signatures of kind of improper processing in some way in the brain and autism that might give us a clue as to where a therapeutic strategy might be developed. So that's the goal. Okay. So we get there by studying transgenic model mice that are engineered to have exactly the same gene mutations as in humans. And because those gene mutations in humans, they model these severe syndromic forms of autism, these mouse models, which are the only things people study, right? These mouse models are not models of high functioning autism at all. So I think very important first, lesson here is let's not mistake anything that we discover in the biology of these mouse models for something that's happening in the large majority of all of our family and friends, right, or ourselves that are somewhere on the autism spectrum. This is not that, right? These are severe syndromic forms. So then your question about interneurons. There's been a theory for this, which is that maybe in the brain, in these severe syndromic forms of autism, maybe the fundamental problem is simple, that there's too little inhibition. There's excitatory neurons, there's inhibitory neurons. Individual neurons receive both excitation and inhibition, and neurons kind of sum them up to decide when to spike, and they produce trains of spikes that encode all the relevant information and perform computations. So the idea was if there's too little inhibition, maybe neurons in the brain, and people have been thinking of cerebral cortex mostly, maybe they're too excitable, they produce too many spikes, and too many spikes would essentially be noise in neural computations. Maybe that's the fundamental problem. Okay, computation. So what we did over a series of studies in my lab starting about 5 years ago is examine that model by looking across multiple different genetic models of autism to look for physiological problems in the brain that are common across the different forms. So we're trying to understand, where are the commonalities across these different forms. And so we found that it is true in many different mouse models that there is too little of a particular type of inhibition called inhibition coming from parvalbumin or PV neurons. So people call this the hypofunction of these PV inhibitory neurons. So that really is true. But when we made measurements to ask whether that causes too many spikes in the brain, the answer was no, it didn't. So brain cells were spiking at kind of normal rates or sometimes even less than normal rates, even though inhibition was low. So we tried to understand what that meant. And we've been doing a lot of measurements recently. We had a paper this past year. We have another paper that's about to be submitted that shows that the loss of this form of inhibition has a different effect on these cortical brain areas. It has a very strong effect of kind of blurring and scrambling the neural code for sensory information. We're studying sensory cortex. So for what sensory information is out there, as if the problem is not in sensory areas of the brain creating too many spikes, which you might imagine might produce a perception of a sensory world that's too intense, let's say. But instead, there seems to be some kind of blurring in the brains of these mice where what's out there is not too intense, but what's out there is maybe blurry and confusing. And so the hypothesis that we're working with now and trying to test now is this true? Is it true that from the perspective of the cortex in these severe forms of autism, maybe there's a blurry and confusing sensory world, and maybe that contributes somehow to the dysfunction that's going on?
Thank you so much for that explanation. That like perfectly covered two of the other questions I had too. I was actually planning to ask about how transgenic mice models allow to make inferences about the human brain, which you covered, saying that it's not obviously a good representation of high functioning autism in humans, and it's not anything to be used as like a perfect biological representation of what we would carry out clinical studies and experimentation to understand.
Exactly. I mean, in any disease model, it's very common to use transgenic mice for genetic diseases. And we always think of them as preclinical models, not clinical models. And preclinical means we're testing ideas about what might work. And the whole goal is maybe there'll be such a strong signature in a mouse that you can develop an intervention, a drug, a gene therapy, a behavioral training, something, right, that normalizes the function in the mouse. And if that's true, then that becomes a candidate that you can try in humans in a clinical trial. And many people are interested in doing that translation step and testing whether it works, but we never can presume that it will work. You always have to test that it will work because it's a real challenge to go from mice to humans. It's unfortunately only a small fraction of the discoveries made in mice that really translate effectively to humans.
Yeah. I just had like it was really interesting to hear about about the research in the field, because like definitely there are like misconceptions, like there are like things like that, like regarding autism in terms of like talking about the spectrum, things like that. I just had like an ethical question regarding also also like when it comes to like a difference between genders, like for like boys, however, it's much autism like phenotype specifically turns out it's like more prevalent, whereas for girls it's not necessarily as prevalent. Like there is that happening, and as a result, there also is they also may women as a result may even get like a possibility to be like misdiagnosed or like not diagnosed like earlier on. Because since early intervention is a really important key factor for autism. So just wanted to like get your thoughts regarding like the ethical component, the ethical ideas behind that, and also like how has the field improved like in terms of like providing an equal diagnosis for men and women, basically.
Yeah, it's a great question. There's many important ethical issues about this. Actually, the top one, even before we get to sex differences, is the issue of neurodiversity, right? Because autism is so broad, it's, in my view, absolutely the case that we shouldn't seek a treatment for everybody because a large number of people with autism, maybe actually the large majority of people with autism, are just representative of the normal diversity of the way the brain works across people. There's nothing abnormal about this. This is totally normal. It makes people unique. Okay. And I think for many types of autism where people have been able to, through the support that they get, be able to successfully attend college, have a job, have relationships, work through life, they're clearly in that set of people. And that's not, those people are not who are doing research to try and cure. In my view, there's nothing to cure there. Okay. But that's very, very different from people at the very low functioning end of the spectrum who have a variety of very, very difficult behaviors where their families and often themselves, if they can express themselves, are desperately seeking some kind of treatment, and there is really no effective medical treatment for autism now. So I think the first important ethical issue there is that nobody is trying to say everybody should be cured. That's absolutely not true. Okay. So you're right that autism is 4 times more prevalent in males than in females. Some of that is probably bias in the diagnostic criteria. I should say I'm not an MD, so I'm not an expert in this. I don't go diagnose people. But many of the features that are associated behaviorally with autism tend to be features that correlate with male behaviors anyway, and so it may be more diagnosed there. And people also argue that maybe in females, maybe because females tend to have sort of stronger social awareness and stronger social interactions, there might be more of an ability to mask those symptoms. And so maybe there's more undiagnosed autism in females as well. So there's many different puzzles to work out there. And I think, in a way, this kind of gets back to the bigger question of why do research in mice, on autism. The fact that we don't know how to segregate the autism spectrum means that we need to understand the disorder better. And I think, myself and many researchers who work on mice, what we're hoping for is that if we study enough different genetic models of autism, they're not going to all be the same, but maybe we'll figure out that there are groupings of them. Maybe there's 5 or 10 of them that seem to have the same physiological problem in the brain, and then a different 10 of them might have a different physiological problem in the brain, et cetera. And there's already hints of that emerging, actually. And if that's true, maybe the mouse work can be used to figure out how to divide up the autism spectrum into genetically similar forms or groups of autism that might be amenable to one therapy for one group and a different therapy for another group, et cetera. And you can imagine that maybe expanding throughout the autism spectrum in a way that really helps people identify more precisely, what's going on with them or their loved ones and help get better treatment. So that's really the grand goal.
So this understanding of, I would say, compartmentalizing different, I guess, like brain changes that are causing autism is not an attempt to reduce the idea of it being a spectrum of autism. It's just a way to provide some more direct clinical implementation of, say, like treatment if there is an issue that needs to be investigated or needs to be treated.
Exactly. We don't call cancer one disease. We call it multiple diseases. And then by studying those different diseases, people have been able over the past 20 years to figure out dramatic improvements for treatments for certain types of cancer. If we don't divide it up into pathophysiological groups, brain mechanism groups, basically, then I think our ability to find therapeutic approaches that work is going to be much more limited.
Yeah. I mean, it's very interesting. Are there other animal groups that could be more similar to humans in terms of like the way the brain functions? If there's a diagnosis of autism other than mice? And my next question would be like, how accessible is that, and how ethical is that?
Yeah, I mean, this comes up for translation of many, many different disorders, right? That before you develop a potential treatment for testing in people, you really want to use it in an animal that's as similar to people as possible, right? And for non-neurological diseases, maybe you want animals that are physiologically similar. But for neurological diseases, you also want animals that may be cognitively or behaviorally more similar, right, in some way. So it is an important step. It does have ethical issues, but I think if they're addressed properly and carefully and regulated correctly, I think that that can be okay to do. And so the species that are sometimes used for this are monkeys, macaques sometimes, or these little primates called marmosets are now being more and more used. It's still relatively rare within neuroscience to do this because it has to be done much more slowly because the animals develop more slowly and there are fewer researchers that work on them. And so it's just a very slow process. So you don't really want to do you don't want to model every one of the 100 genetic forms of autism in monkeys, right? That would be crazy and really, really not a good use of monkeys. You want to do some screening to understand it in simpler organisms. And then, in my view, when you have, a couple of approaches that seem very, very promising, you might want to, if possible, look at them, in primates before looking in humans. There are some cases where it goes directly from mouse to human as well. So you don't need to use primates, but that might be a possibility.
Okay. That's interesting how you say that. Like it can sort of almost be like another level before you reach the clinical thing. Yeah. We just like to all say that we really appreciate your time. Just sort of like as a closing question, like are there some very like pressing like myths or misconceptions that like you hear like I don't know like on TV in the news that are like spouted about the brain, that you just like to correct?
Like myths, brain myths.
Yeah, I mean,
Yeah.
Just like yeah.
Um, I don't know if I have a particular brain myth. I mean, I'll tell you one idea that gets discussed a bit that I personally don't agree with. I think is probably wrong. That is this idea that if somehow you could create a model of your own brain and upload it to the internet, that's where your consciousness would be and you would live forever. And I think that is, a sort of science fiction romantic kind of thing to imagine. But I think we are really tied to the biology of our brains, you know, and as it stands right now, you know, if we're lucky, our brains will be in good function for 100 years. And 100 years is a long time. We should enjoy that. We should treasure every moment of it. We should enjoy each other. We should support each other. And then when the 100 years is up, I think that's what we got. I don't think
Yeah.
uploading your consciousness to the internet will help.
Thank you so much, Dr. Feldman.
Okay, you're welcome. Take care, you guys.
You as well.
Yeah, thank you.